we have a really fantastic talk from Dr. Susana Carmona on neuroanatomical changes associated with human pregnancy. I will say that although we have the X symbol there, we're really on Blue Sky now, and I will be trying to post on Blue Sky during the talk, so you can always follow me there. Um, Before we really begin, I'd like to recognize that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. And I also acknowledge that many of you are not joining us from Canada, and I encourage you to learn about the Indigenous peoples who occupied the land before you occupied the land. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, which is covered by Toronto Purchase number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members also live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. And this is run by the Women's Health Research Cluster. Who are we? We have over 800 members across 32 countries. It's free to join, so please join us if you're not already a member. We host a number of events, such as the seminar series that you're listening to right now. And we work across diverse fields. We're not just researchers or trainees, we're also stakeholders and community members. And I think now more than ever, perhaps it's really important to keep the engagement up in the international community because I would say there's a little bit of an anti interest in things like women's health, and we need to keep the pressure, I think, on the system. And you can see a number of our collaborators. Please become a member. And we have a new website, which is really fancy. So now I'm going to stop sharing. And while I stop sharing, I'm going to invite Dr. Carmona to share her screen while I read out her bio. Now, we edited it down because it's extensive.
Oh, just sum it up, sum it up.
I'm summing it up. I'm summing it up. Dr. Carmona is a senior researcher, and I'm not going to say this properly, Instituto de Investigación Biomédica del Hospital Gregorio Marañón. I'm sure I didn't do that very well at all, but she's in Madrid, Spain, where she directs a neuroimaging laboratory. It's called Neuromaternal, which specializes in studying the human maternal brain, and we'll hear more about that. She co-directs a number of groups in Spain, but also internationally, including the Pregnancy and Motherhood subgroup of ENIGMA and the Maternal Brain Project at the Anne S. Bowers Women's Brain Health Initiative. Her scientific contributions include over 59 peer-reviewed publications, including a seminal one in Nature Neuroscience, which I'm sure you'll hear about. And she's won numerous awards. Over the past decade, she has really laid the foundation for her laboratory's research looking at the longitudinal effects of pregnancy on the maternal brain. And as many of you know, I like to give fun facts of our distinguished speakers, and Dr. Carmona has just given me a plethora of fun facts. She loves to paint, which is her passion. I don't think this is one of her paintings, but it's beautiful
I wish, I wish. No, no, no. This is Sorolla. This is
And
Sorolla.
when she has insomnia, I hope it's not too often, but she likes to listen to true crime podcasts to help her fall asleep. Now, I don't know that everyone would choose true crime podcasts to fall asleep to,
That's
but
what I thought it was a fun fact because I don't even understand why. But I guess my mind is even worse than a crime.
no, it's great. Thank you so much for sharing those. And I look forward to your presentation.
Thanks. Thanks a lot, Lisa. Thanks for the nice presentation. It is, of course, a pleasure to be here presenting at this Women's Health seminar series. But I have to admit that it feels kind of strange to be invited by you to give a talk about the maternal brain when you have been a reference for me during many years. So please feel free to interrupt me to enrich whatever I'm gonna say. Okay. I think I told you many times, but I'm very conscious that you and many others have paved the way for us to advance into the neuroscience of human pregnancy. Okay. I always like to start by the take-home message, because I would like you to know by the end of this talk that we have solid evidence demonstrated that the transition to motherhood entails profound, dynamic, and long-lasting changes in brain anatomy. And also that we have preliminary evidence that suggests that these changes seem to facilitate maternal-infant bonding and seem to be mediated by gestational hormones, especially sex steroid hormones. So my idea during the rest of the talk is to present the studies that support this take-home message. Okay, so let's start. As many of you already know, pregnancy is probably one of the most extreme physiological adaptations a human being goes through, because in just 9 months, almost every organ in the woman's body adapts to facilitate gestation, parturition, and postpartum. We have huge changes at the cardiovascular level, at the respiratory level, at the musculoskeletal level. The immune system switched from pro-inflammatory to anti-inflammatory. So there are all these changes that affect our body. But pregnancy is not just about adapting or changing the organs that we already have. Pregnancy also entails the creation of a new organ, which is the placenta. And the placenta, that is an endocrine organ, synthesizes and secretes a series of molecules that travel through the bloodstream, hormones that travel through the bloodstream, and bind to specific receptors in the woman's body, informing the rest of the organs how they should function and communicate during this special status. They tell the mother's body that now everything should work under a different set, under a different range of parameters. So everything works okay to gestate, give birth, and then the postpartum period. Well, so we have a lot of medical manuals describing in details all these peripheral changes and what to do in case something goes wrong. But we're just beginning to understand how pregnancy shapes the brain. And this is especially concerning for several reasons. And I'm going to just mention 2 of them. One of them is that we know that approximately 1 out of 5 women will suffer postpartum depression, that we know it's a very debilitating disease and impacts the well-being of the mother, but also of the child. And in fact, we also know that suicide due to mental health problems is the leading cause of mortality in the United States. This is one of the reasons. And the other reasons is that outside the context of motherhood, we have a lot of evidence demonstrating that these hormones are powerful neuromodulators. We know that these hormones bind to specific receptors in brain cells and induce a series of neuroplastic changes. And these neuroplastic changes go from changes at the molecular level that affect the synaptic transmission between neurons to changes in the morphology of brain cells and also changes in the number of brain cells, not only neurons, but also glial cells. And as you can see in this graph, pregnancy is like the quintessential hormonal storm. There is no other period in the life of a human being in which her body and her brain is invaded by all these powerful neuromodulators. So just to give you some data, the levels of estrogens accumulated during the 9 months of pregnancy are equivalent to those accumulated in a non-pregnant woman during approximately 35 years. Despite all that, as I have mentioned, we are just starting to understand how these pregnancy hormones shape the human brain. We barely have any evidence from humans, but fortunately, we have a lot of evidence from non-human animal models, thanks in part to Lisa. And from these 50 years of evidence in non-human animal models, we have a clear relation between gestational hormones, neuroplastic changes, and maternal care. So I'm going to just briefly dedicate a couple of slides to review what we know from murine models, and then we go back to humans again. So from murine models, especially rats, we know that gestational hormones such as estradiol, progesterone, oxytocin, and prolactin bind to specific receptors in the mother's brain, in particular in a region known as medial preoptic area of the hypothalamus. And from this, 2 main things happened. First, there is an inhibition of the avoidance or regression circuit, and in parallel, there is an activation of the reward and motivational circuit, this mesolimbic dopaminergic circuit. So once the first is inhibited and the second is activated, the pups become a very powerful, salient, and motivating stimuli for the mother, who will search to be in contact with the pups. And is this contact, this interaction with the pups, which will further shape the brain of the mother and adapt maternal behavior to the changing demands of the pups. And we also know that this maternal care can also be induced in virgin female and male thanks to the continuous exposures to the pups. But this takes a little bit longer, like around a week or so. So in mice, researchers have managed to refine so much that they no longer talk about the medial preoptic area of the hypothalamus. They can talk about a specific set of neurons known as galanin neurons, that are basically neurons that express a neuropeptide galanin. And what these researchers do is to use fiber photometry recording and optogenetic activation to demonstrate that these neurons are critical to parental care by suppressing parental aggression and enhancing parental motivation. I don't have time to show you the videos, but I really recommend you take a look at them, the videos that are linked to this publication here, because it is amazing to see how researchers can turn on and off maternal care just by manipulating this set of neurons. And very recently, on November last year, the group of Johannes Koll demonstrated in a very cool study that estradiol and progesterone produce specific effects on these neurons, so that these galanin neurons respond persistently, strongly, and more selectively to PAP stimuli. So for instance, estradiol seems to salient some of the noise of these neurons to enhance those that respond to PAP stimuli, while progesterone seems to rewire the circuit, so these changes are long-lasting. Well, if you want to know more about the details, the neuroplastic mechanisms that accompany pregnancy and postpartum in murine models, I recommend you reading this review we published last year, where Camila Servin and Magdalena Teresa did a great job synthesizing the work of giants in the field, such as Lisa Gallia, but also Jody Pawlowski, Benedetta Leuner, Joseph Lonstein, and so on and so forth. Okay, so this is a very short synthesis of what we know from animal models. But what about humans? Is there a relation between gestational hormones, neuroplastic changes, and maternal care? Well, surprisingly, until very recently, we didn't even know if that was true. We didn't even know if the human brain will change during pregnancy. In other words, in 2017, we didn't even know if the brain of a newly produced woman will change once this person becomes a mother. So this is what I try to address in my first publication as a co-senior author, and I'm going to tell you about this seminal paper. What did we do here? What we did was to contact women that were planning to get pregnant but didn't have any children, and scan them before they got pregnant, and then again at 2-3 months postpartum. And as control groups, we included not only newly produced women that do not have child and do not plan to have children in the future, but also their respective couples. So we have 4 groups: first-time mothers, first-time fathers, and then newly produced couples. And I'm going to show you the results of comparing the brain at these 2 time points. This is what we found. So as you can see, we don't find any difference in the rest of the groups, but in the mothers group, we detected very significant gray matter volume reductions. And these reductions were not randomly located in the brain. If you see this image, you can detect that these reductions were confined to what we know as the full mode network. That is a large-scale functional network that is involved in internal processing, mind wandering, self-referential thought, and also theory of mind and empathy. We can discuss about that later if you want. Okay, so once we found these results, we were wondering how long do these reductions in gray matter volume last? Are they something specific of the early postpartum, or do they persist for a longer period? And to answer this question, what we did was to contact a subgroup of participants, a subgroup of these 25 participants, and scan them again at 2 years postpartum. And here I show you the results. So each of these graphs is a region of interest, and in blue, I have shadowed the pregnancy period. So you can see that there are reductions from preconception to 2-3 months postpartum. And for most of the regions, these reductions persist at least at 2 years postpartum. Okay, so this is a very schematic representation of what we found. We found, again, gray matter volume reductions from preconception to 2-3 months postpartum that are still detectable at 2 years postpartum. Once we saw this, we were wondering, what do they mean? Are they related to cognitive performance, what it's commonly known as mommy brain or mommynesia? Well, in this study, we also collected a set of metrics that assess verbal memory and working memory, and we did not find significant differences between the group, nor we found any association between the brain changes and the variability in cognitive performance. What we found were associations between these brain changes and scores on maternal attachment scales. So we found that brain changes can predict the scores on maternal attachment scales. And this association, together with all the literature from non-human animal models, suggested to us that perhaps these brain changes might facilitate maternal behavior. Again, if you want, we can discuss about that later, because I know it might be a little bit controversial. But this is what we found. Okay. So this study for me answered a very important question, at least from my point of view. And we demonstrated that, yes, pregnancy produces gray matter volume reductions that persist at least 2 years postpartum and that seem to facilitate maternal attachment. However, what is even more important is that this finding opened a series of questions that we, as well as other researchers in the field, you can see some of the names here, have been trying to answer during the now 7, nearly 8 years. And what I'm going to do during the rest of the talk is to try to answer to some of these questions, not all of them, because I think I won't have time. Okay. So the first question we had was this one. Are brain changes similar to those taking place during adolescence? Why did we ask that question? Well, we wanted to answer these questions because when the results of the first study that I told you about reached the press, it had a great media coverage, and some of the journalists understood the findings, but others made statements such as the ones you can see here, right? So we found like headlines with sentences like this one: woman's brain atrophy with pregnancy. And when we had the chance to respond, we usually tell them that a reduction in gray matter volume does not necessarily imply a neurodegeneration, and that in biology, as in many things in life, sometimes less is better. And to respond to that question, we usually use the example of what happens during adolescence. And we explain them that during adolescence, there are these big changes in sex steroid hormones that seem to produce different cellular changes, such as synaptic pruning and myelinization, that at the macroscopic level are translated into reductions in gray matter volume, and that these reductions in gray matter volume are concomitant with cognitive development. So those regions that are in charge of more sophisticated processes are the ones that show this gray matter volume reduction later in life. This is what we told them, but we hadn't empirically tested if the gray matter volume reductions that we found during pregnancy were actually similar to those that occurred during adolescence. And we did so a couple of years later in a publication and in human brain mapping. And here what we did was to scan a group of girls before and after puberty and compare the brain changes, the anatomical brain changes in this group of adolescents with those that take place during pregnancy, again, before conception and 2-3 months postpartum, and again, including a control group. And for this study, instead of just looking at gray matter volume in general, we use surface methods that give us a very detailed picture of the anatomical changes. So we can quantify whole area, cortical volume, surface area, cortical thickness and a set of indexes of gyrification. And I'm going to show you the results here. So on the y-axis, you can see the monthly rate percentage of change. And on the x-axis, you can find all these metrics that I told you about: total brain volume, cortical thickness, surface area. And in green, you see the controls, which are near the 0% change. And as you can see, the adolescents in red and the mothers in blue follow a very similar pattern of neuroanatomical changes. Okay. Does that mean that pregnancy and adolescence entail similar changes also at the cellular level? We don't know, because we have to remember that we're looking at the brain at the voxel level. And within each voxel, we have hundreds of thousands of brain cells. So different changes at the cellular level can produce similar changes at the voxel level, similar signal changes at the voxel level. But I'd like to think about these findings as a kind of proof or support to this beautiful term name called matrescence that was coined. I don't know if you heard about that. Lisa, have you heard about matrescence? Yeah. So this was coined by the medical anthropologist Dana Raphael in the 70s, and it refers to the process of becoming a mother. And I like this word because it kind of merges motherhood and adolescence to highlight the parallelisms of these two life stages. And one of these parallelisms is to see this process as a neurodevelopmental one rather than a neurodegenerative one. But that's just my wish. Okay, I'm going to skip this question due to time constraints, and I'm going to jump directly to the next one. Do brain changes reverse? Again, let's recover our schema. So we have that when we compare preconception measures to 2-3 months postpartum, we found profound decreases in gray matter volume that are still present at 2 years postpartum. Okay, so this means that at least they last 2 years postpartum for sure. But then we have another set of studies, like large-scale cross-sectional studies that compare thousands of women that were mothers to thousands of women that were not mothers. Those are data from the UK Biobank. This is the studies of Anne-Marie Delange. And what they found is that even at these ages, those women that were mothers show a younger estimate brain age compared to non-mothers. And this finding has been replicated by other researchers, such as Edwina Ocker, and this has been observed also not only at the neuroanatomical level, but also at the functional level or even the cognitive level. And what this data is suggesting is that pregnancy might have a lifelong impact on the brain. But of course, this is evidence from cross-sectional data. Ideally, what we might need is to have longitudinal studies that compare or that follow the same individuals across time. And this is what we tried to do in another publication. We contacted the participants from the first study in 2017 and scanned them again at 6 years postpartum. And the first problem we had was that the sample was very small. Why? Because some of the pregnant women already have a second child between the sessions, and some of the controls, including myself and Elselina Huxema, became mothers during this period. So the sample is very small. We have 7 mothers and 5 nulliparous controls, but so far it's the only data set that follows participants from preconception to 6 years postpartum. And here I show you the results. So at the top, we have the comparison of the initial study, Huxema 2017. And there you can see these reductions in the full mode network. And at the bottom, I display the results from comparing the preconception to 6 years postpartum, sorry. And as you can see, some of the regions of the default mode network, such as the medial prefrontal cortex and the precuneus, are still reduced at 6 years postpartum. Okay, so to the question, do brain changes reverse? The answer for now will be no. At 6 years postpartum, some of the volumetric reductions in the full mode network are still detectable. Next question. Do fathers undergo similar brain changes? So, like 15 minutes ago, I showed you this image, and I told you that we didn't find any difference on the father's group and that we only detected differences on the mother's group. But the truth is that when we compare the results, the differences that we detected in the mothers were so pronounced that we couldn't believe them. So we have effect size of nearly a Cohen's d of 2, which is very, very large. And in order to be kind of sure that what we were finding was robust, we use a very restrictive threshold at expenses of missing some positive findings. And using this restrictive threshold, only the mothers group show significant differences. But when we plot the raw data, we found distributions similar to the ones that you can see here. So you have cortical volume, cortical thickness, and surface area. And as you can see, the distribution of controls, which is the blue one, is centered in the 0 of no change. The mothers, which is the pink one, is squeezed to the reductions, and the distribution of the fathers kind of lays in between the two. So some of the fathers change, but the changes are not so strong as the ones we found, or so pronounced as the ones we found on the mothers. So all in all, this makes us believe that some of the changes we detected on the original study might be driven by environmental factors related to childbearing. Well, I don't have time to go through all of these publications, but very briefly, we found that we explored this data further, and we found that changes in fathers seem to be associated with the father's brain response to infant cues, to fluctuations in oxytocin and testosterone, and to parental engagement. And we have replicated some of these findings into international database. If you want to know more, the leading researcher in this field is Darby Saxbe, so you should check her publications. So to the question, do fathers' brain undergo similar brain changes? The answer will be yes, but the changes are smaller than those detected on the mothers. And in fathers, neuroanatomical changes seem to be associated with parental engagement. Next question. Which is the temporal course of these brain changes? Is it linear, quadratic, exponential? Okay, so far, again, our schema here. So far, I told you about the studies, the longitudinal studies that compare preconception to postpartum metrics. And these studies are consistent in demonstrating that there are reductions associated to pregnancy. But there is another set of studies that compare the brain of mothers at different time points during the postpartum. And what these studies found were the opposite. They found increases in gray matter volume. So how do we fit all this data together? Well, by pulling all this data, different trajectories emerge, right? So it is possible that what it's happening is that there are decreases during pregnancy that seem to valley around birth and then slightly recover, but do not reach pre-pregnancy levels. But this one was also possible, right? So maybe the reductions that we were attributing to pregnancy were in fact produced merely by birth. And in order to answer this question, what we did was to perform a study that zoomed into the peripartum period. And in this study, what we did was to scan 110 pregnant women at the 3rd trimester, and then during the first month postpartum, some of them as close as the first week postpartum, and as controls, because MRI is very noisy, we always have to have controls. As controls, we included 34 control women scanned at similar time intervals. And to be completely sure that our findings were robust and reliable, we included a replication sample of 29 pregnant women scanned at similar time points and controls. And this subsample was scanned at a different scanner and at a different city. And I'm just going to tell you the results that replicate across these two data sets. So the first thing we did was to cross-sectionally compare these two groups. Cross-sectionally compare these two groups. And what we found was that at the 3rd trimester, the brain of pregnant women was already reduced in cortical thickness, which is suggesting that these reductions were present before birth. And we also performed this longitudinal comparison, and we found that these initial reductions attenuate as postpartum progresses, right? And the more the percentage of postpartum time between the sessions, the more the reductions or the attenuation, which is suggesting a trajectory compatible with this one of decreases during pregnancy that seem to valley around birth, slightly recover during the early postpartum. But one thing that surprises a lot was that we were expecting only reductions in the default mode network, as in our initial study. But when we compared the groups, we found that these reductions were widespread. They were not confined to the default mode network. So we performed additional analysis at the network level to try to understand this incongruency. And what we found was that those regions that fall into the frontoparietal on default mode network seem to recover at a lower rate. And perhaps this is what we detected, only differences in this region at 2 months postpartum, and then at 2 years postpartum and 6 years postpartum. Okay. So if you're wondering how consistent these findings are, I can tell you with a high level of confidence that this trajectory is highly consistent. So consistent that it can be detected at an individual level. So a couple of months ago, the group of Emily Jacob published this wonderful study in which they used then sampling to follow a single participant, Elizabeth Crustil, during her first pregnancy, and they scanned Elizabeth 26 time points during gestation and up to 2 years postpartum. And again, what they found was decreases in cortical thickness that seem to have their inflection point around birth, and then slightly recover, but without reaching preconception levels. And what is interesting is that these studies also measure diffusion, and they found, for instance, that global anisotropy has a different trajectory. So global anisotropy is kind of a measure about the white matter integrity. And they found that in the case of global anisotropy, the trajectory was different. It was characterized by increases up to the second trimester and then decreases later on, and again, do not reach in pre-pregnancy levels. So what this is indicating is that we urgently need a multimodal characterization of the human maternal brain, because only having this map of how the brain changes, we will be able to identify potential deviations, such as the one that might take place in perinatal disorders such as postpartum depression. So to the question, which is the temporal course of the brain changes, the answer will be that it depends on the brain metric, but for gray matter, it seems to be characterized by decreases during pregnancy that valley around parturition and slightly recover in the early postpartum, but do not reach preconception measures. Next question, are these changes mediated by hormonal fluctuations? We have a lot of evidence from urine models demonstrating that this is the case, that gestational hormones shape the anatomy and the functionality of the brain and facilitate maternal care. But what about humans? So we recently published a paper in which we scanned 127 gestational mothers and have them have an MRI at preconception, 18 weeks gestation, 32 weeks gestation, and 1 and 6 months postpartum. And as control groups, we included not only the nulliparous women, but also a very interesting group of non-gestational mothers that are basically the same-sex couples of some of these mothers. And I'm going to show you the results here. In green, the nulliparous women, in blue, the non-gestational mothers, and in orange, as you can see, the mothers, the gestational mothers that show this pronounced U-shaped trajectory. And in fact, if you can see here, I don't know if you are able to read that, but we found that at the peak, there is around a 5% decrease in cortical gray matter, which is a lot. And in this same study, we also found that the trajectory of gray matter volume changes was associated with the trajectory of 2 metabolites, sex steroid metabolites, which is estradiol 3-sulfate and estrone sulfate. So those women that have more changes in gray matter cortical volume are those that have also more changes in these metabolites. And again, here we found an association between the gray matter volume and maternal attachment scales, and we found that this association was mediated by maternal well-being. So to the question, are these brain changes mediated by hormonal fluctuations? The answer will be yes. Glomerular volume changes seem to be mediated by estrogen levels. And of course, as we advance in the field, more questions arise. So for instance, as I have mentioned, what happens in white matter integrity, in resting state? Is there a relation between all these brain changes and immune factors? How do these changes relate to other important peripheral markers, such as microbiota or vascular changes? Are these brain changes different depending on breastfeeding or parturition type? What happens on second pregnancies? Can we use this data to kind of predict and prevent postpartum depression? And more general questions such as, are these changes different depending on the genetic profile, race, ethnicity, gender, socioeconomical status, parental leave policies, so on and so forth? You can imagine probably you also have additional questions. So we recently wrote a comment whose first author is Magdalena Martinez, and there we were like presenting the most pressing questions in the field of human pregnancy and proposed the use of a dual approach. On the one side, we propose to use dense sampling precision studies, such as the one that I told you about, in which they scan a single woman during 26 time points. And on the other hand, on the other side, to use large-scale consortium studies. So there we can have the benefits of these two approaches. For instance, dense sampling studies are ideal to capture within subject variability, and thus they are going to be ideal to capture phenomena that are highly dynamic and the association between them. So for instance, to answer the questions at the top. And large-scale cohort studies will be ideal to capture between subject variability and can help us to answer the questions at the bottom. So of course, if we really want to advance in the neuroscience of human pregnancy, we need large-scale prospective multimodal database, and this is what we have been working on together with a group of Oscar Villarroya. We have been building a large and comprehensive longitudinal database of first-time mothers that now includes around 400 women. Some of them have all the time points, but not all of them. And these women have an exhaustive information about neuroimaging data, including T1, T2, resting, and diffusion. We also have some biological samples, clinical and neuropsychological tests, as well as behavioral paradigms. And they have been evaluated before conception, at mid-pregnancy, late pregnancy, early postpartum, and up to nearly 2 years postpartum, 1 year and a half. But we need to go even bigger and scale from the order of hundreds to the order of thousands. That will be ideal. And therefore, we are collaborating with two very important international initiatives, such as the ENIGMA-Neuroendocrinology and the Ernst Bauer's Women's Brain Health Initiative. And I want to take the opportunity of being here talking to you today to invite you to look up more information about these two initiatives online. Okay, so just to warm up, I'm going to finish with a take-home message, because I hope I have provided enough evidence to demonstrate that we have now solid evidence indicating that the transition to motherhood produces profound, dynamic, and long-lasting changes in the anatomy of the human brain, and that we have preliminary evidence indicating that these changes seem to facilitate maternal-infant bonding and seem to be modulated by gestational hormones. Well, that's all from my part. I want to thank all the authors that you can see the names here, and especially the ones you can see here at my laboratory. If you have any questions, I'll be more than happy to answer them.
Well, thank you so much, Dr. Carmona. I know it's really hard to do these virtual talks, so if the participants can do some like a little emojis or something so that you know, we can hear it. We can't really, but we can see it, and I see a number of them coming through. I hope you do too. So thank you very much. I also see some very familiar names in our audience, which is fantastic. please feel free to ask a question. You can put it either in the chat, which I have not been monitoring, or or you can raise your hand and we will get to you. And in the meantime, I can also obviously start asking some questions. So I think like such a tour de force, like force, not purse, force of data is so incredible. And that hats off to you for doing such a great job in presenting it. Um, it's interesting. This is a minor comment, but I think some of the first inclination is to think that fatherhood or even same-sex partners wouldn't have it would be an environmental cause if you see any changes in brain. But hormones can also be involved, and I think you pointed out to oxytocin and testosterone. Is there any work in looking at even same-sex um, uh, uh, parenthood, like non-birthing parents, to see if there are hormonal changes there as well?
Yes, Darby Saxbe have done really cool job on fathers, and I know that James Rilling, and I'm not sure if this data is already published, but he's also
Yes.
exploring that on fathers. And I want to say that, I mean, what we are seeing, sorry, I'm gonna be here.
Oh, it's
What
okay.
we are seeing are like changes, and some of them are related to maternal behavior, but that there are no changes on the brain does not mean that. I mean, those changes kind of are associated with maternal care, but they don't determine
Yeah,
maternal
yeah,
care.
that
So,
was my
so
next question. Yeah, yeah, yeah.
yeah, I know. I have to punctualize this because, especially in non-scientific audiences, it's like the proof whether you can be maternal or not. I mean, it's obvious that adoptive mothers are great mothers, right?
Yeah.
And it's also obvious that these changes might not only be directed to maternal care. There are many other things that we want to discover that may be behind these huge changes.
Well, of course, like because there's no other questions yet, so I'll better say it. I'm very, of course, it makes sense in the rodent literature that they looked at maternal behaviors first, right? Because that's very salient behavior that changes. And for those of you that don't know, the rats and mice are actually neophobic to new pups until they become a mother, or they have those hormones, or they have the experience with pups. Then they'll show those changes, those maternal behaviors, like licking, for example. Like we all lick our babies. And but we and others have shown pretty profound changes in the other regions too that are not associated with maternal behaviors, like the hippocampus. And I know you also see changes in frontal cortex and hippocampus. So I know you didn't see any changes with in terms of memory, but what do you think that possibly could be involved in?
I think there is something we need to explore better with more profound batteries. I'm not sure if the batteries that are
Mhm,
designed to
mhm,
capture very big differences, like
mhm,
for instance, to measure changes on persons with dementia or something like that, are subtle enough to capture this feeling that many mothers have about um, problems focusing or something like that. And I know that this is a very complex phenomenon. There is these expectations, there is this mental load, there are many things to take into account, but
mhm,
I think that it's necessary that we develop comprehensive cognitive batteries to see if some of the changes might be um, related to this, and if they are, kind of know what to do with that. Can we treat them? Can we somehow
mhm,
control what is going on?
mhm,
Um, yeah, we found some difference on the hippocampus,
mhm. Yeah, I know. I
we
use
also
it all the time in my talks. So thank you for that.
yeah, and I think that well, it's also interesting to explore how these brain changes in the hippocampus relate to the memories they have during birth, and we're also
Mhm,
exploring that, like
mhm,
this
mhm,
birth experience and how this might impact the hippocampus.
mhm. Yeah.
But it's a whole field to discover, and I think we have a lot to do there. And I know that we have to look at all the data you have published on murine models to have some case of how to advance there. But on humans, we don't have a lot of data about the potential I mean, when we measure them longitudinally from preconception to postpartum, we haven't found any associations with cognitive metrics with the
No,
budget.
no, yeah, and the cognitive measures that you're using.
Mhm.
Also, there's a question in that, but before we get there, in terms of the long-term changes, like again, in terms of hippocampal neurogenesis, so this is different, but you do see them suppressed for a long period of time, and then they come up. And I kind of think about Henry DeLange's data in the same way because so I know you didn't see them. You still see the reductions in grade matter at 6 years postpartum, but hopefully you can keep following those individuals and see whether much later in life, like at age 60, for example, you still see those changes because that's more, I think, I know it was a UK biobank, so I think those individuals on average are much, much older in terms of when
Mhm.
they were scanned, right? I think that it's very possible you won't see the sort of
Mhm.
decrease in brain aging or that sort of
That's our
until
dream.
later. Yeah.
Yeah, that's our dream, to create a large-scale longitudinal
Yeah.
database and see how gestation impacts menopause and see
Yeah.
the
Yeah.
long-term effects.
Yeah.
And actually, we haven't focused on the hippocampus on the 6 years follow-up study. We just
Mhm.
focus. So maybe
Yeah.
there's something
Potentially.
interesting there,
Yeah.
but we only focus on the regions of interest, the
Well,
default network.
you also showed, and hadn't seen that part, but the default mode network and the frontoparietal changed less dramatically. Which ones were the ones that changed a little bit more dramatically in the early postpartum?
No, they changed more
Oh,
dramatically.
they changed more dramatically,
So
but
in this
mm,
study, we found, so on the first study, we only have this pre-parturition and post-parturition, and what we
right,
found was like the recovery rate was different. So
right,
those that affected the hormone network and frontoparietal regions
yeah,
decrease at a lower rate. But in this study that I just showed you, this one that is on the screen,
hmm
we also measured the network level, and we found that those that have more pronounced reductions are those that affect higher order um,
hmm
large-scale functional networks, such as the frontoparietal and the default. And the one that is more preserved is the limbic one,
mm,
which
interesting.
is really cool. Yeah.
Yeah.
Mhm.
There is a question in the chat, which is, I wonder why this question was asked, and I don't know if do you want to unmute yourself and ask it yourself, or you want me to read
No,
it?
no, no, go ahead.
Okay. Does pregnancy change pain perception and pathways?
Super cool.
Mhm.
Yeah, I
Yeah.
always want to do that because I don't know where I read that somehow
Yeah.
in an anesthesia manual or book, like
Yes.
a very old
Yes.
one. And I've been trying to convince a friend of mine that does pain to try to
Yeah.
collaborate with that and do some kind of pain perception study. It's a
Well, I
super
have
cool
to tell you,
question.
anecdotally, it changed my pain perception.
Oh,
It's like
yeah. And
I
part,
thought
yeah,
I thought I knew
yeah,
what pain was, but then having had a natural childbirth twice,
yeah. You had a natural one?
yeah, both times, yeah. It was so much fun. Um, uh, Another question. Curious about the mothers versus fathers. Do you find any one of them had more variability than the other? So was there more variability in one sex versus the other in terms of
Very good question. So I'm going to plot the results of the bootstrap distribution. We don't have enough data. I usually said that there is more variability
mhm,
because, I mean, because if I can say that some of them change and some they not, but
mhm,
the distributions are not extremely different in terms of variability. You can see them here.
mhm,
But probably we have more data that will make sense because it will be consistent with the fact that maybe fathers parental care or brain changes are related to engagement, and there might be more variability. There's something to study at different cultures and societies to see.
mhm. Laura Gravelsons has a question too, which I think is one of the questions that you gave to at the end, which is I'm wondering if you find trajectory of brain changes for first-time pregnancy is similar for second-time pregnancy. And if you haven't looked at this yet, do you have any hypotheses about what might happen?
I think that there will be also very pronounced changes. We are exploring that now. I haven't analyzed the data. We are collecting now second
Okay.
pregnancies.
Yeah.
So in terms of the brain changes, I think that a very similar trajectory will happen, maybe not as pronounced, but in terms of maternal care, I know, and Yulisa can can enrich this answer, that in murine models there is this prime first pregnancy, and then later on maternal care it's like facilitated once you have been a mother for the
Yeah.
first time.
Yeah.
So that's also another possibility, that these huge brain changes are more pronounced during the first pregnancy and then just subtle adaptations um, during the follow-up
Hmm.
pregnancies.
Um,
But we
yeah.
haven't empirically tested that yet.
Yeah. We have a little bit of rat data on that.
Mhm.
But mice and rats are quite different in terms of their maternal care, so that's also kind of interesting. Um, Romina Garcia de Leon has a question, which is, can you tell us what sort of biological markers you'll be evaluating in your current project?
Yes, so we want to do proteomics
Yeah.
to kind of have a sense of what might be going on in terms of the immune system, all the peripheral changes that I told you about. And also we are acquiring microbiota, and basically we want to extract all the fluids we can. But
Yes.
I mean, there is like this trade-off between making the woman feel comfortable during the procedure.
Sure.
But yeah, the idea is to measure peripheral markers, not only hormones, but also epigenetic markers and proteomics.
Hmm. Yeah. There's Romina on the screen right now.
I see her.
Hi, nice to meet you. I just
Likewise, Romina.
I actually have like a weird question because I'm not super familiar with neuroimaging. Is it common to do those kind of replication studies at other, no, it's not. Well, I thought that
No,
was
we are super picky with
impressive.
that.
Yeah.
Yeah. And we can do that here. We are so sure that these findings are so pronounced that it's like an extra preview. Sometimes you just find something and it's like, okay, it's significant, but sometimes it's in the edge. And here it's like, look, this is real. There's no
Yeah.
way, and I'm gonna show you that this is something that I can find here. Can Eveline can find the same in Holanda, in the Netherlands, sorry.
Mhm.
My colleagues in Barcelona can find the same, and even in California. This is something that it's real. And this is very unusual in neuroimaging. This is very unusual.
Wow. Well, congratulations.
No,
That's
thanks.
amazing.
Thanks, Romina.
Thank you. Nice to meet you.
You might see her at Parental Brain in Barcelona.
Yes.
And then there is another question which I also have asked before in the past, but do you see any relationship with sex of the baby? So does it matter? Do you see different changes depending on male or female?
I remember we have a very nice review that asked us that
Yes.
on the
I
first
wondered who
study.
that was.
And I don't know why, but I always thought it was you, and now
Yes,
I
it
think it's
was
you.
me.
No, we haven't found that. So
Yeah.
it doesn't mean that maybe there are, but so one thing that I want to highlight here is that the changes are so big that there might be smaller changes
Mhm.
that are overshadowed
Mhm.
by all these enormous changes. So maybe we just focus on this time point and use a
Mhm.
more lenient threshold. There are subtle differences, but not comparable to this huge
Mhm.
curve. And we don't find difference between the sex of the babies. We don't find differences between twins and not twins and
Mhm.
single tones.
Mhm.
That's how you said that, single
Yeah,
tones or
singletons. Yeah. Interesting. That's also interesting. What about
it's
adoptive parents?
we are also acquiring adoptive parents now.
Mhm.
And I think they will follow a trajectory similar to that on the non-gestational mothers.
Mhm. And one of the questions I had, and then you showed some data, I think it's from Emily Jacobs' lab, the dense sampling, but does it also change? Do you see similar changes in white matter?
We haven't. So the thing is that on this first study, the
Mhm.
within pregnancy, we didn't
Mhm.
acquire measures of diffusion. We just acquired before conception and then after parturition. Now we are acquiring measures of diffusion weighted images during pregnancy, and we will be able to see how reliable, how replicable this trajectory are on other metrics.
Mhm. Fantastic. Um, yeah, it's what New Maria, is that how you say your name? I also had the very same question, so I love it, of course. Um, if we don't have any other questions, are there any other questions? I am going to invite um everyone to give another round of thanks to Dr. Carmona for an excellent talk and a really fantastic overview of everything that might be occurring in the terms of the neuroscience of the maternal brain. It's really important work, and we're really, really glad that you're doing it. So thank you. It took too long for somebody to do it. And so thank you so much for all your efforts. It's great.
Thanks to you, and especially to you, Lisa, for inviting me to give the talk.
No. Thank you for agreeing. So thank you.